Let's take a quick overview of Chapter 13, which covers microbes, fungi, and protists. How do we split up these major groups? One of the things we do when we're splitting up these major sections of the tree of life is we ask whether or not it's a prokaryote or a eukaryote. What that means is, do they have a nucleus? Eukaryote means true nucleus. Prokaryote is before the nucleus. Prokaryotic cells do not have a nucleus. Everything in the blue and red is prokaryote. Another way that we can look at it is whether or not they're multi versus single-celled. I have a question mark here because this one is a little bit tricky, but for the most part, protists are more or less single-celled organisms. Whereas if we look at plants, fungi, and animals, those typically are multicellular. The other thing we look at is a cell wall present? And if so, what is it made of? Everybody in these two groups will have cell walls. The difference is, what is it made of? This area over here in blue will have a different type of cell wall than this whole area here in red. We also see cell walls in fungi, composed of something different. We also see cell walls in plants. Once again, different composition. Next, we can look at the mode of nutrition. Are you an autotroph, meaning you are able to create your own energy using inorganic compounds? You will find autotrophs, of course, in the plants. You'll find some autotrophs here in our protists. If we look at animals and fungi, those are heterotrophs. Animals ingest their foods. Fungi are heterotrophs, but they absorb their food. Rather than bringing the food in and then breaking it down, they secrete their enzymes on the outside and then they absorb that across their body. These are the important things you really have to know where to place animals along this tree. Let's take a look at the tree of life here and look at the groups that we're going to talk about in this video. So these are our prokaryotes. Within the prokaryotes, we have the red group there are the archaea, or sometimes called archaebacteria. Where you see them placed, they actually are a little bit closer to the animals, plants, and fungi because of some of their other similarities, which we won't go into. Then we have the bacteria, sometimes referred to as the eubacteria. We have two different groups here of prokaryotes, archaea and bacteria. In this video, we'll also talk about the protist, so we can see this protist grouping over here. And then we'll also look at the fungi. Early life on Earth was hard. The atmosphere did not have any free oxygen in it. It was anoxic. It means that early life forms had to be able to do anaerobic respiration. Anaerobic means in the absence of oxygen. They had to be able to perform metabolism without using oxygen. It wasn't until later that photosynthesis and aerobic metabolism, which means in the presence of oxygen, evolved. Another thing about early atmosphere was there was a lot of strong radiation. Early life forms were what we call extremophiles. They lived in these really extreme environments, and they still do in many of those groups. Deep ocean floor where there's hydrothermal vents below the Earth's surface, because they were essentially avoiding that strong radiation. Part of what occurs when you have this challenging life is you see the formation of biofilms. And essentially it's like a little community that forms of these microbes, and they interact with one another and they essentially help one another in a lot of ways. So there's a lot of mutualism going on there. Biofilms have become very important because in, let's say, hospital equipment, biofilms will form on the surface of some of this equipment, and it can be very difficult to clean equipment that has biofilm on it. So it's something that is actually very relevant in today's world, not just in early Earth. Bacteria come in three basic shapes. We have the round shape here is called coccus, rod shape is called bacillus, and the spiral shape is called spirillus. And then you also can get them either in individual units or they can form chains. So if you see something called streptococcus, it means it's a chain of these spherical cells. Now, a prokaryotic cell will all have certain features. They will all have a plasma membrane, which is the same as the cell membrane right here. On the inside of that, you'll have sort of this semi-solid, semi-fluid type of a consistency called the cytoplasm. Inside, you'll have a genetic material. Remember, this genetic material doesn't have a nucleus around it. And then you also will have these ribosomes. Ribosomes are for protein synthesis. And ribosomes are a little different than some of our other cellular organelles because they aren't membrane bound. So those are the general characteristics that prokaryotic cells will have. And then they'll typically also have a cell wall. Depending on the type, it'll be a slightly different cell wall. Remember we said cell walls are important. We break the tree of life down into do they or do they not have a cell wall, and then if they do, what's it made of? For the bacteria, there's two different types of makeup of the cell wall. They both have peptidoglycan. So one of the key features is they have a cell wall made of peptidoglycan, whereas the archaea have cell walls that are made of a variety of different compounds, but not made of specifically this peptidoglycan. And then we can separate the types of bacteria into the structure of their cell wall. And based on this structure, they will stain differently when you do a gram stain. So if you have gram-positive bacteria, the peptidoglycan is all here on the outside, whereas over here in gram-negative, this peptidoglycan is sandwiched in here between an inner and an outer membrane. This one stains negative, it's a gram-negative, and then this one stains positive, gram-positive. So it's where that peptidoglycan is sitting that makes the difference. Let's talk a little bit about the modes of reproduction of bacteria. One of the things bacteria can do is this first one called transformation. And what we see in transformation is this is some DNA that's just kind of floating around in their environment, and they pick it up. They pick it up and they incorporate it into their own nucleic acid. And whatever genes are located on them, they now have those genes. One of the reasons why bacteria evolve resistance so easily is they pick up this stray DNA, and sometimes that stray DNA has ampicillin resistance and other kinds of resistant genes out there. Another way that we can get new DNA into a bacterial cell is through transduction. This little hexagon structure here is supposed to represent a virus. This virus has nucleic acid in it. It's going to inject that into the bacterial cell, and that bacterial cell is going to incorporate it here into its own DNA. And then finally we have conjugation. In conjugation, one bacterial cell will give some of its DNA to another bacterial cell. Now, some prokaryotes are harmful. You've heard of MRSA, methicillin-resistant Staphylococcus aureus. So these are bacteria that have become resistant to methicillin antibiotics. We also see that E. coli can contaminate food sources. Salmonella can also contaminate food. And botulism can cause disease. Now, botulism is kind of an interesting one. It can, under certain circumstances, be used beneficially. We can use the botulism toxin to create medications. For instance, migraines are often treated using Botox injections in areas where there's a lot of overactive nerves, and it causes those nerves to quit sending all of those pain signals, and it helps reduce the frequency and occurrence of migraine. So we also have other beneficial prokaryotes. Some are used, of course, to make various food items, cheese, yogurt. Many species of bacteria can also break up and degrade oil. In some big oil spills, like the Exxon Valdez spill, we can release these kinds of bacteria to break that down. We refer to it as bioremediation. We are using a biological organism to remediate. We also have a lot of beneficial microbiota in and on our bodies. We have a lot of beneficial microbes in our gut that help us digest food and works with our immune system. We have a lot on the surface of our skin. So there's a lot of good bacteria. And part of the problem with overuse of antibiotics is not only are we allowing things like MRSA to evolve because we are essentially creating a very strong selective pressure for those that can get around our antibiotics, but we are also, in many cases, killing off our good bacteria. That's one of the reasons why sometimes when you take an antibiotic, it's good to, at the same time and afterwards, take a probiotic to reintroduce good bacteria into your body. We have the prokaryotes, we're going to move into eukaryotes. Where did they come from? Now remember, one of the things that we find in eukaryotes is we have that membrane around the nucleus. We also have these other organelles that have membranes around them. And two of the really important ones are chloroplasts and mitochondria. because these are the energy harvesting organelles inside of cells. Chloroplasts perform photosynthesis. Mitochondria perform aerobic cellular respiration. How did they arise? And so this is known as the endosymbiotic theory. We have essentially the precursor to a eukaryote. We have a prokaryotic cell. One of the first things that's going to happen in this prokaryotic cell is this membrane is going to start folding in on itself to create some of this endomembrane system that we see here. And then we're going to have an endosymbiotic event. What that means is this cell is now going to engulf another cell. First, it's going to engulf an aerobic bacterial cell. So this is another prokaryote that is performing aerobic metabolism. Essentially, that cell now becomes the mitochondria that we see inside of these eukaryotic cells. This cell then also can do a second endosymbiosis and ingest a photosynthetic bacteria. This photosynthetic bacteria is essentially going to give rise to chloroplasts. Here we would have the origin of our organisms that perform photosynthesis. Here we have our origin of organisms that do not perform photosynthesis. But both of these are now eukaryotic cells. Remember, we said when we get to eukaryotic cells, we're going to have sort of two major kinds of groups. We're going to have those that photosynthesize and those that are heterotrophs. They don't photosynthesize. Let's go over to protists. So protists are eukaryotic. They have these membrane-bound organelles. What are some of the other characteristics of protists? Well, I put a big question mark here because protists are tricky. Protists are basically the junk drawer of taxonomy. It's probably not a true single group. So when we make a group in taxonomy, we infer that that entire group has a common ancestor. And that probably is not true for protists. They are very diverse. Their structure is diverse. You can have anything from single-celled organisms. You can have these very large multicellular kelp beds. They're very different in the way they reproduce, different in the way that they obtain energy. So some are photosynthetic, some are heterotrophs. So they are just all over the place. Now, one of the things that protists do, if they are heterotrophic, they have to ingest food. This is showing how they ingest food. This is called phagocytosis. They're taking their cell membrane and pushing it out into like these little arms, pushing the cytoplasm out into it to wrap around this food particle. Or if this is one of your white blood cells, this may be debris, damaged tissue. It may be a pathogen. It's going to wrap around that, and we're going to create what we call a food vacuole. And then inside this cell is another sac that contains a bunch of digestive enzymes. And that is going to merge with the food vacuole, and that's going to allow this cell to break that down. It's going to use what it can, and then whatever it can't use, it'll essentially do the reverse of this phagocytosis. It'll come over here and it'll eject what it cannot use. Now this shows us the supergroups for the protists. Again, keep in mind that different books will show you different things because it really has not been resolved. We do not know exactly how to resolve the protist group. Some protists are pathogenic. Up here we see the organism that causes malaria. This Plasmodium, it's italicized, so that means it's a genus name. And when you see this SPP, it means that there's a variety of different species that can cause malaria. Rather than list them or identify them as individual species, we'll just say it's several different Plasmodium species, and that's how we'll do it. Trypanosoma, several different species can cause African sleeping sickness and Chagas disease. We also have some protists that are pathogenic to plants. We see our downy and powdery mildews on plants are caused by protists, and we also see the potato blight is caused by protists. Now, some protists, of course, are also beneficial. They form significant components of our food webs. A lot of plankton are protists, and they form the basis of the food web for aquatic environments. Protists account for about 25% of the world's photosynthesis. We also see that protists can be symbiotic organisms to others. Here we see an example of coral. Inside the coral, you have a photosynthetic protist that is living inside the coral. It's providing energy for the coral, and the coral is providing a place for that protist to live. You also see symbiotic relationships between termites and protists. Termites do not have the enzyme for breaking down cellulose, which is what wood is. They have these symbiotic organisms that live inside their guts that do that for them. And then, of course, a lot of protists are living in the soil, and they act as decomposers, returning dead and decaying organisms, releasing those nutrients back into the soil. What are some of the characteristics of fungi? I put this here again so we can see where the fungi are over here. So we're definitely eukaryotic because the only things that are prokaryotic are the red and the blue here. All of the brown are eukaryotic. They are typically multicellular, although yeast are considered fungi and they are unicellular. They are heterotrophic, so they don't do photosynthesis. They have to consume their food. Now, one of the differences between animals and fungi, they're both heterotrophic. When animals consume their food, they ingest it and then they break it down. When fungi consume their food, they break it down and then they absorb it across their body. That's why we say here they acquire their nutrients by absorption. And then another difference here is that they have cell walls, and so we're going to see plants have cell walls too, but their cell walls are made up of chitin. So it's not the peptidoglycan that we saw in bacterial cell walls. It's not the type of compound that we see in plant cell walls. That's going to be cellulose. It's a different compound. It's called chitin. Chitin is the same thing that an insect skeleton is made of. So let's take a look at the basic structure of a fungus. The fungus has a vegetative body, which is essentially everything down here. And then there's the reproductive part, which is also called the fruiting body. Now that vegetative portion is made up of these individual strands that we see here called hyphae. And then if we have a whole mass of them, imagine just sort of a matted network of these, it's called the mycelium. And the hyphae we see blown up over here. The hyphae can have actual septae between the individual cells, or they might not. So this could all be one huge cell with lots of different nuclei in it, but they're not separated by a membrane. Here they are. Let's take a look at the major divisions of fungi. We have club fungi, mushrooms you're used to, sac fungi, mycorrhizal fungi, we have the conjugated fungi, and we also have chytrids. And then this is just showing where animals fall in relation to the fungi. Some fungi are parasitic or pathogenic to plants. This green mold on grapefruit, we can see fungus growing on grapes, powdery mildews and rust. We also have parasitic and pathogenic fungi on animals. Ringworm is a fungus on the skin. We also see fungus that causes mycoses. Mycoses is just a disease that's caused by fungus, and we see that on the scalp. We see a fungus that is creating these little white dots, these little specklings here in the lungs. So it's a fungal infection in the airways. This is a type of fungal infection underneath the skin. Some fungi, of course, are beneficial. Mycorrhizae are very beneficial fungi. They create these mutualistic symbiotic interactions with the roots of plants, and it allows the roots of the plants to have a better uptake of nutrients. So they're showing here that if we have more mycorrhiza down here, the plant is growing better. It really increases the health of the plant. A lichen is actually a symbiosis between a fungus and an algae, and they have different forms. You can see them growing on the surfaces of rocks and trees. A lot of our fungi are decomposers, working down there in the soil to return those nutrients into a usable form for other organisms. Fungi can be used for pest control. We can introduce fungi to control for, like, say, crop pests, and some fungi are used for food. Here we see a morel mushroom, which is a type of a sac fungi, and then we also see a truffle, which is a type of a club fungi. So that gets us through chapter 13.